Okay, welcome. Thank you everybody for coming to this session. It's the last of the originally scheduled sessions, but Sean just informed me that there might be more coming. So if you do like this session and you have somebody that in mind that might want to see it, I am recording it, so it will be posted on the library website in their LibGuides. And there might be some more scheduled sessions coming up in the future. So hello, my name is Corey Alexander. I have been teaching anatomy, physiology, microbiology, and biology at RCC for about a long time, about 15 or 16 years. 16 years, going on 17 this year. So I was approached actually in January, it seems like a year ago, to do something like this for faculty, just to present some information. That's when the vaccines were first coming out. And so I've just modified this a little bit with updated with some of the new information. And now we're offering it to anybody, RCC, faculty, staff, and students who want to learn a little bit more about how their immune system works and the specific COVID-19 vaccines that are available. So this is going to be a little bit of a science lesson to understand how our cells work and how our immune system works. And then we'll finish up taking a look at specifically how the currently approved, fully approved and emergency use authorization approved vaccines in the United States work. I am happy to pause, take questions, just let me know. You might have to speak up if I don't see hands or faces or anything, but there is going to be a time for questions at the end. So it's very informal. If you need me to slow down or repeat anything, just let me know. All right, I think I'm ready to go. If I can, there we go. So I'm going to start off with a little bit of background information regarding our cells. So if you've had cell biology or maybe just high school biology way back when, you might remember some of these things. I'm not going to focus on everything of the cell. I'm going to focus on just the components that are play a role in how the vaccines work and kind of our immune responses. So what you see on the screen is a typical animal cell. The key parts that we are going to be taking a look at, I almost used my finger. I don't have a touch screen at school. I have a touch screen at home, is the nucleus, okay, and all of its different pieces. The nucleus is that big purple blob in the middle. The nucleus is a structure inside of our cells that contain our DNA. So they're calling it chromatin here. But chromatin is just a fancy word for kind of loosely coiled DNA. It's basically all the instructions for how your body works. It's the blueprints, you may have heard it be called. So it contains the genes that codes for all of the cell products that you need to survive and for your cells to function. The other thing that we're going to take a look at is this rough ER, stands for endoplasmic reticulum. That rough ER is just a big network of membranes, plasma membranes, that are studded with these little organelles called ribosomes. Okay, so the instructions for all of your cells' properties are held within your DNA in the cell's nucleus. And then the rough endoplasmic reticulum and these ribosomes are what I like to call the workbenches, or they are the place where the products, the instructions are stored in the DNA. The products of those instructions are manufactured on the ribosomes. There's a whole bunch of other fun parts in the cell, but we're not going to get to that. We're just going to focus on those components. So the nucleus with the genetic information and the ribosomes and the rough endoplasmic reticulum. Okay, so now we're going to zoom in to a close-up of the nucleus, taking a look at that DNA and a few of the processes that take place on a day-to-day and minute by minute. These are happening in all of our cells right now. So here's our nucleus. We kind of have this barrier here. All of our DNA is held within this nuclear envelope. And so this blue ribbony looking double helix is the DNA. And the way that our instructions are kind of organized on our chromosomes or in our DNA is in these small little sections called genes. So let's say from this point here, I'm not the best at drawing with my mouse, and this point here. So everything in between those two red lines, you have this sequence of these colored bars. These are called bases. A's, T's, C's, and G's. That may or may not sound familiar to you, but it's the sequence of these bases. What is the order of those letters, A's, T's, C's, and G's? That specific order is the instructions for what we call, or that sequence is what we call a gene. And that gene codes for a protein product, right? So an analogy that I often use in my classes is making cookies, right? So usually when you make cookies, you have to have a recipe, and then you follow the recipe and you make your cookies. But let's say you have a grandma that she's got like her world famous cookies, and she's very covetous of her recipe. So she just doesn't give out her recipe cards, but she's going to give you permission to come in and copy that recipe onto your own recipe card. So this is a process here that we call transcription. Kind of, we can see it up here on the top. Transcription is the process in which your DNA instructions in this gene get made into a temporary copy called RNA. And specifically, it's called mRNA. That M stands for messenger. It brings that message of the instructions for that protein from the blueprint of the DNA. We're transcribing it into this temporary copy. So that'd be like you going to grandma's house. Her recipe stays locked away, but you get this temporary copy, and she says, you can have this as long as you promise to rip it up and destroy it once you're done making your cookies. And you're like, okay, grandma, I promise. So here we have this piece of mRNA. It has all of the instructions of DNA for just that one protein product from that one gene. In a real life example, if we don't want to talk about cookies, that could be like the hemoglobin protein that's found inside of your red blood cells, or that could be the insulin hormone that your pancreas releases to help regulate your blood sugar. It could be a receptor protein that's found on the surface of all of your cells, like your red blood cells for your blood typing, if you're type A, B, those types of things. Those are all protein products, and they're coded for by a single gene, usually. So what happens to this transcript? Well, in our cookie analogy, you're going to take your recipe and you're going to go home and you're going to follow the instructions and make the cookies. Well, in a cell, our kitchen, if you will, is going to be those ribosomes attached to that rough endoplasmic reticulum or free floating in the cytoplasm. This is the site of where your proteins are made. And so what happens is, as that mRNA goes in and finds a ribosome, lands on the ribosome, what you'll see is some other types of RNA called tRNA, and they're bringing in all these little circles. These circles are called amino acids. They are the building blocks of proteins. So a protein is a big, long chain of amino acids, and it folds and it twists into a very specific three-dimensional shape, and that shape gives it its ability to do its job, whether it's being insulin or hemoglobin or receptor protein or some other protein in the cell. So as this ribosome kind of reads the sequence of letters in the mRNA, it brings in the correct amino acid from the cytoplasm and hooks them together into this protein, and that is your product. So that would be you adding the sugar and the flour and the chocolate chips and mixing and then baking it, and now you have your cookie. That's your protein product. So the instructions are stored at grandma's house. That's your DNA. You got your temporary copy. That's your RNA. And then you go to your house and you make the cookies, and that's your protein product. So this is going to be really important when we come back and take a look at how the vaccines work, because they take advantage of how our cells naturally make proteins to allow us to make some of these viral proteins. Okay, so that's our cell biology background. The next piece we're going to take a look at is your immune responses. How does your body fight off foreign invaders? Whether it's COVID or flu or chickenpox or E. coli, whatever the pathogen, the disease-causing agent is, we have built-in ways to fight that off. The first ways that our body can fight off pathogens or disease-causing things is what we call by innate or non-specific immunity. So I have this collection of just little pictures, and these diagrams are coming straight from the books that our students are using when they take my A&P class. So I'm just going to briefly go through these. These are defenses that we are born with. We don't have to have any prior exposure to anything for them to do their jobs. So the first one, the most obvious one, is our skin, right? It is great. It helps keep us wet on the inside and keeps us dry on the surface. We have glands that secrete things like sweats and oils that help keep our surfaces protected from pathogens. And it's intact, right? It's dry, and it's got these thick layers of dead cells that keep us from bacteria and viruses getting deeper into our tissues. But it works best when it's intact. If you've ever had a cut or a scrape, you know that increases the chance of infection. And what's infection but bacteria and viruses coming into our deeper tissues? So intact skin will fight against any pathogen that's trying to get into our bodies. The next group are cells called phagocytes. Phagocytes means cell eaters. Basically, they're cells that can go around and gobble up things. They can gobble up pathogens like bacteria. They can gobble up dead cells or debris, waste products just from your day-to-day activities. So they can play a role in protecting us from pathogens by eating the pathogens and getting rid of them. And they'll do that against any particular type of pathogen they come across. The next type is what we call immunological surveillance. It uses a cool group of cells called natural killer cells, which I think is an awesome name. Basically, what they do is they roam around like a surveillance, like a police crew, and they roam around and they can recognize if any cell is showing an abnormal, like a name tag, like, hi, I'm Corey, I'm a normal Corey cell. But if one of my cells is abnormal, either it's a cancerous cell or maybe a virus has infected it, its name tag is going to be a little bit different. Instead of saying, hi, I'm healthy, normal Corey cell, it's going to say, oh, I'm sick, I'm infected Corey cell. So these natural killer cells roam around your body looking for those abnormal signals. When they find those, they can actually directly contact, they come and touch that cell, they release chemicals and destroy that cell. So it doesn't proliferate and cause problems inside your body. And again, it will do that against any type of abnormal cell. The next two groups are kind of chemical protection. We have one group called interferons, which are chemicals that cells release when they're infected by viruses, protects their neighbors from infection of that same virus. The next group is called complement. Complement is a series of proteins that are found naturally in your blood plasma. When they get activated, they destroy bacteria. So we've got these two kind of chemical weapons, so to speak. One targets viral infection, one targets bacterial infection. The next is our inflammatory response, inflammation, right? If you've ever had a cut or a bruise, it gets hot and warm and red and really like tender. That's inflammation. It's a normal part of our immune response. And then lastly, we have fever. If, think of the last time you have a fever. This is a good thing up to a certain point because our cells can withstand a wider range of temperatures compared to pathogens. So bacteria and viruses are very narrow in their ability to replicate inside of our bodies. So if we crank up our thermostat a little bit to say 101, 102, 103 degrees, our cells can handle that and they actually do pretty well, but bacterial replication, viral replication goes way down. So it helps us get other parts of our immune system to come in and get a leg up on this infection that we have. So that's a quick, whirlwind tour of what we call innate or non-specific defenses. These are things that are going to work no matter what kind of pathogen is coming into your system. Okay. The next part of our immune response is what's called adaptive or specific immunity. Now, this is what we're trying to trigger when we do vaccines or when we actually get sick in response to something, is these responses, the cells that are involved in our specific immunity are programmed to recognize just one pathogen. So if we've all, I don't know the age groups of the people who are watching today, but chickenpox is a very, people know what chickenpox is. So you have either most likely been exposed to chickenpox in the past when you were a kiddo, or maybe you were in the age group that you were able to get the vaccine when that came out. Either way, you have cells that have been programmed to recognize just chickenpox. They ignore flu. They don't have anything to do with COVID. They can only work when it has something to do with when they're recognizing chickenpox. So that's when we talk about adaptive or specific immunity. You have to be exposed to this pathogen. Then you get these cells that are programmed specifically to recognize that one pathogen. Okay. So within this arm, this specific defenses, your adaptive immune defenses, we have two groups of cells, or two groups of cells. We have T cells that are responsible for what's called cell-mediated immunity. And then we have B cells that are responsible for what's called antibody-mediated immunity. So in cell-mediated immunity, we have T cells that can either directly attack an infected cell. In B cells and antibody-mediated immunity, the B cells produce antibodies that can target the pathogen that's outside of cells. And on my screen, I don't know if you can move the little strip of faces, but there's a cartoon over here that says B cells, I attack invaders outside of the cells. And they do that by producing antibodies. T cells say, I attack infected cells because they have the ability to recognize when a cell has a pathogen, like a virus or a bacteria inside of it, and can kill it before that cell releases those newly formed viruses or bacteria. Okay, so we're going to take a look at cell-mediated immunity first. I'm going to move all my people back over here. All right, so in this particular diagram, we're taking a look at a group of T cells called cytotoxic T cells, which basically means cell-killing T cells. They're not the same as natural killer cells. These are cytotoxic T cells. So in this diagram, what we see is this nice big yellow cell, this big blob here. This is, say, one of your body cells. Maybe it's a throat cell. We can, COVID or influenza or chickenpox, whatever. And it's being infected by a pathogen. In this case, it's showing both viruses and bacteria. So these are the bad guys. They've come into your cell. They've invaded your cell. So let me step back. In a natural, healthy cell, we are constantly putting up little name tags. These are little proteins that our body produces that say, hey, I'm Corey cell, I'm healthy. I'm Corey cell, I'm healthy. But when you get infected, some pieces of those pathogen, whether it's the viral pieces or a bacterial piece, they get processed and they get incorporated into those daily name tags that say, instead of saying, hey, I'm healthy Corey cell, it's going to say, hey, I'm infected. I'm an infected Corey cell. And this is a piece of that thing that's infecting me, whether it could be a viral spike or a surface protein on a bacteria or whatever got processed. It gets placed up onto the surface of the cell. And now it's sending a signal, say, hey, look at me, I'm infected. Come and kill me before these pathogens have any more chance to replicate inside. Okay, so when a cytotoxic T cell comes around, so that's in the first panel there, you have your infected cell. It's waving its I'm an infected name tag up there. That a cytotoxic T cell has been programmed specifically to recognize that one bit of pathogen that's been exposed. So in the case of COVID, it's going to be the viral spike. So if you get infected with COVID, your cells are going to they're going to get pieces of that COVID virus and put it up on that name tag saying, hey, I'm infected with COVID. It's going to take some time because COVID's brand new to the human population, but you're going to get these cytotoxic T cells that have been programmed to recognize just that viral spike. When they do, they become active. That's the middle panel. They go through many, many rounds of division. So they're producing a whole army of these cytotoxic T cells that can recognize anything, any cell that's showing that COVID spike name tag. A small group of those cells kind of are dormant. They're called memory cells. They do not fight in this current battle. They stay off to the side for later. And then once your T cell, the cytotoxic T cells become active, it will then destroy that infected cell. It goes in, recognizes that name tag, releases chemicals, kills a cell, very similar to the natural killer cells, but it's specific to just that pathogen that's causing the infection. Okay, so I have a micrograph of an actual kind of process of these cytotoxic T cells destroying their target cell. So on the top, on the left-hand picture, there's a smaller cell on the top, has come into contact with a large infected cell on the bottom. It's recognized it, it's going to release its chemicals, and in the bigger picture in the middle, we can see what that infected cell ends up doing is it basically gets destroyed. It gets holes popped in the membrane and all the insides just kind of spill out and it's dead. So if you had a pathogen inside of that, like a virus trying to replicate or a bacteria trying to replicate, we've destroyed that virus factory. And so this is how these cytotoxic T cells go around with precision and destroy the cells that are infected, showing that I'm infected name tag that we saw earlier. Okay. The other types of cells involved in our cellular immunity, cell-mediated immunity, are what we call our helper T cells. So helper T cells, they're also programmed to recognize a specific pathogen, just like our cytotoxic T cells, but they don't destroy cells. They are like our managers. They have the ability to release chemicals and manage all different parts of your immune response. So instead of recognizing that name tag on an infected cell, that's just cytotoxic T cells. Remember those phagocytes, those cells that go around and kind of munch up and eat pathogens? Well, when they do that, they have a different name tag. They put out this label that says, hey, look what I found. Look at this bacteria or virus that I've engulfed, and I'm going to put that up on my surface, and that's going to be recognized by the helper T cells. Okay. So when a helper T cell comes in and recognizes that signal from the phagocytic cell, oh, I recognize you. I'm going to become activated. You're the good guy, so I'm not going to destroy you. They get activated, they go through many rounds of division, they produce a little population of memory helper Ts, those go off to the side. And what you'll see on the bottom is those activated helper Ts just start releasing all of these chemicals. Those are the red arrows, these cytokines. And those cytokines can go and enhance your non-specific defenses. So it can activate fever, inflammation, your interferon, your complement, your macrophages. It just kind of beefs up that non-specific defense. And then those cytokines will also go to help activate the next part of our immune response, which is the B cells. Okay, so that's what we're going to take a look at next. So B cells, again, these are cells that are programmed to recognize a specific pathogen. They tend to hang out in our lymph nodes. So if you've ever had swollen nodes like up in your neck or your armpit, those are your lymph nodes. B cells hang out in there amongst many other places in our body. So in this particular diagram, in that step 1, the antigen is a little red circle. So that could be a virus spike, or it could be a bacterial protein, whatever it happens to be. It's recognized by the receptor on that B cell. It's processed, so it kind of takes it into the cell and puts it back out on that name tag saying, hey, look what I found. That is recognized by that activated helper T that we just saw on the last screen. And then that activated helper T then activates the B cell, right? So they're like a partnership. Once the B cells become activated, they go through lots of rounds of division, making a whole bunch of themselves. A small little group stays as memory cells to fight later and years down the line. But then those activated memory cells change their makeup from just B cells to what we call plasma cells, which if you take a look at the drawing, it should look like they're jam-packed full of that rough endoplasmic reticulum. And they are. It's the site of protein synthesis, and antibodies are proteins. So there's going to be genes that code for these specific antibodies. These plasma cells spew out like hundreds of millions of antibodies per hour once they get activated. And those antibodies flood your blood, your lymph, all over your body. And what they do is they can bind to the pathogen and basically neutralize it. So here is kind of a picture, a close-up of what those antibodies can do to protect our cells. So normally, so this red spike here, this is what's called the spike protein. Its job, as far as the virus is concerned, it will come in and it will bind to this receptor. That's just a normal receptor that's found on our cells, especially in our respiratory tract. And when it binds, it tricks that human cell into letting the virus come in. They hijack, they're sneaky. And so if that viral spike can bind to a receptor, that's what triggers infection of your cells, and the virus can start replicating and making more of itself. Well, if you have all of these antibodies flooding around your body, binding and recognizing that spike protein, you've basically blocked the ability for the virus to bind to the receptor. And then we basically cut off that ability for the virus to be infectious. It can get into our body, but it cannot get into our cells. And the only way viruses can replicate is they have to get inside of our cells. So antibodies just take out that whole ability for the virus to be infectious. So that's our little mini lesson on how our immune system works. So we have innate kind of built-in responses, and we have those really powerful specific immunities with our T cells and B cells. So now we're going to take a look at immunity in general, some of the properties of our immunity and vaccines. So the first time we're ever exposed to something, we typically call this the primary response. So that would be for everybody on the planet. If you get COVID, that's your primary response. Nobody's ever had it before, but it could be the first time you've been infected with a particular strain of the flu or a cold virus, or the first time you got chickenpox. Our bodies respond, everything we just talked about. These particular graphs are going to focus on antibody production. So in that primary response, it might take a week or so, maybe even 2 weeks before those antibodies get high enough to actually do something in our bodies to neutralize all of those viruses that are circulating around. So they're showing two of the most common types of antibodies. They're called IgG and IgM. So they peak about 2 weeks after your primary exposure. Remember all those memory cells? We had the memory helper T cells, the memory cytotoxic T cells, and the memory B cells. So let's say you fight off whatever this infection is. We'll say chickenpox, right? So as a kid, you get chickenpox, you get all this itchy spots, then you recover. And a couple years later, your little sister gets chickenpox. And it's not like, and you don't get chickenpox, which is great, but it's not like the virus doesn't come into your system. You live with your family. Chickenpox is very contagious. But what happens is when chickenpox does come into your system, those memory cells are there ready to recognize it and fight it first thing. So take a look at that secondary response. This is going to be 3, 5, 10 years later, depending on the pathogen and your immune cells, that IgG from day one, it's that right at the very beginning of that timeline, it shoots way high. So all of these antibodies, these B cells, those memory B cells become activated. They produce this huge response and they take out that virus before it has a chance to even make you sick. Okay, this is what we want for vaccines. The primary response is triggered by the vaccine in a safe and controlled manner. The secondary response could happen when you get exposed to COVID naturally, that we want that really fast, really high level response before it even makes us sick. So that's the goal. That's how our immune system works, not just with vaccines, but every time we get exposed to something, if we've seen it before, that secondary response comes in and we may not even feel any signs and symptoms, and it just wipes it out before it makes us sick. Okay. So then with these different types of immunity, the purple box, that's our non-specific, our innate. We're not going to talk about that. The adaptive, again, it's specifically your body's response to a particular pathogen. So there's a couple different groupings of immunities that we're going to take a look at here. So on this left-hand side, these are all what we call active immunities. So active, it means your B cells have been recognized a particular pathogen. They are making the antibodies. Okay, passive is different than that. So then how do we get our B cells to make antibodies? Well, there's a couple different ways. If we get sick, I'm not the best at writing with a mouse here. If we get sick naturally, right? You know, little sister sneezes in our face. Our B cells are going to be activated and they're going to be making their own antibodies in response to that exposure. Okay, that's called naturally acquired active immunity. We're getting it from a natural scenario. The next one is artificially induced. So this is what we talk about when we get a vaccine. I'll just abbreviate there. So when we get a vaccine, we are artificially exposing our bodies to this pathogen or this protein spike, whatever it happens to be in the vaccine. And then our B cells are activated to make these antibodies. So it's still active, but it's artificially acquired because we are purposefully exposing ourselves to that particular pathogen in a vaccine. This other group here, this is called passive immunity. So in passive immunity, it is somebody else is making the antibodies and we are getting those antibodies to protect us temporarily. This does nothing for long-term protection. It is as long as somebody else's antibodies are in our system, we get protection. Just like with active, there's two ways we can do that. Naturally, think of breastfeeding and developing fetuses, right? Moms B cells are making the antibodies. Those confer protection on the developing fetus. And then as long as the baby is breastfeeding, those mom's antibodies kind of umbrella that baby and protect it from whatever mom was exposed to in her life until the baby starts getting exposed to its own pathogens, and then it will go to its naturally acquired active immunity or getting its vaccines. The other type of passive immunity, the artificially acquired, this would be like what's called convalescent plasma. I'll just abbreviate. Convalescent plasma is, so let's say you've had COVID and you've recovered, you can go and donate your blood, and your blood plasma has anti-COVID antibodies circulating around in it. So they can extract your plasma with all of your anti-COVID antibodies, and they can give that to somebody else who's sick with COVID. Your antibodies go and fight off their virus. It's not going to make the sick person's B cells activated. That's already done by the naturally acquired virus that they're fighting, but your antibodies are helping somebody else. This is artificially induced. So we are injecting somebody else's antibodies into your body to give you a temporary kind of an extra layer of protection. Okie dokie. Okay, so a little history story. This is always an interesting story to tell. I talk about this in my microbiology class. We're talking about the history and the development of that science. So Edward Jenner is depicted here as the gentleman in the picture. He was making some observations way back in the 1700s. Smallpox was still running rampant, but there was a similar disease called cowpox. Not as dangerous, not fatal at all, but he was noticing the cow maids, the women who their jobs were to milk the cows, they had nice smooth skin. Smallpox tended to leave lots of scars, so you could tell if somebody had survived smallpox. And so he's like, well, how come all of these cow maids seem to not have any smallpox scars? Is there something about them that protects them from getting sick with smallpox? So he made some more observations and kind of decided to do this experiment, totally unethical now, but this was a long time ago. He inoculated his gardener's son with cowpox. So he basically on purpose made the kid sick with cowpox, let him recover, get better. Then he purposefully infected that same child with smallpox. The little scabs off the smallpox blisters are infectious. They have the virus in them. And so he basically made the kid exposed to smallpox. And luckily, even though he had no idea how it was working, because they didn't know about antibodies and B cells and T cells in the 1700s, the boy did not get sick with smallpox. And he did it multiple times. He kept, exposing this child to smallpox and he never got it. So he kind of concluded that if you recover from something similar, you will have some level of protection. And so this kind of kick-started the research into vaccinations. And Louis Pasteur did some vaccinations on rabies and some other diseases back in, again, 17 and 1800s, a long time ago. But as that science progressed, even up into the mid 1900s, so we can see kind of these charts for polio, rubella, measles, and mumps, some of the very common childhood diseases that were, pretty widespread in the United States. When these first vaccines started to roll out on a large scale, we can see how successful they were in reducing the overall cases of these 4 diseases in the United States, almost down to 0. Not completely, they're not eradicated. Smallpox is the only virus that's ever been eradicated in a wild population. There's still polio and rubella, mumps and measles out there, but it's not anywhere close to where it was before vaccines were available for these childhood diseases. Okay. So what is a vaccine? Well, a vaccine is a piece or some representation of that pathogen, whether it's a viral spike or a surface protein or a bit of that, like the identity, it's like the name tag of whatever that pathogen is, that we're trying to induce that specific immune response. We're trying to get those B cells and T cells ready to recognize it. So then if it comes in the future, we'll be able to fight it off with that secondary response. Traditionally, right, so some of the new COVID-19 vaccines are more of a new version. So traditionally, there was 2 major ways that vaccines were produced. You have what's called live attenuated. Get my pen up here. So live attenuated means you have a living pathogen. even though I'd argue viruses are alive, but it's infectious. So you have this living pathogen, and what they do is they culture it. They grow it over hundreds of generations, and they select for the strains that still have the identity, I'm COVID, or I'm influenza, or I'm, chickenpox, but those viruses can't make you sick. So they're attenuated, they're changed in some way. And it usually is by selective breeding, if you will, selective culturing of these viruses and bacteria. And so the benefits of a live attenuated virus is you get a really strong immune response, because to your body, it looks like, hey, this is a naturally occurring bacteria or virus, but it can't make you sick. The downside to that is it's still considered a living, reproducible organism, and there are chances that it could back mutate into an infectious version, even though that doesn't happen. That's still a possibility. And it's a lot harder to produce. It takes a long time to do those culturings to find that strain that still has the, hey, this is me name tag, but can no longer cause cells to get infected and cause symptoms of a sickness. The other group is called inactivated. And in this case, they take the bacteria or the virus, whatever the pathogen is, and they kill it with different ways. They can kill with chemicals or radiation or heat. And they break it up into pieces so it's dead. It can no longer be infectious. And then they take some of the pieces of that virus or bacteria and they put that into the vaccine. And the presence of those pieces is what's going to trigger your immune response. So benefit is it's cannot cause disease. There's no way it can make you sick because there's no living reproducible part in that vaccine. It's easy to produce and it's easy to store. The downside is you get a little bit of a weaker response because your body's like, it's just a bit, it's not the full thing. So typically you get boosters with these types of inactivated vaccines. So I have a list of some of these traditionally produced vaccines that you may have familiarity with. Either you have them yourself or you've had kiddos that you've got their childhood vaccines. So there's quite a few in both categories. So quite a few of the attenuated, quite a few of the inactivated vaccines. Okay, so this is when before the COVID-19 mRNA viruses came out, these were the two ways that vaccines were produced. Okay. In this process of vaccine production, the FDA has a very specific timeline on how vaccines are produced. So you're going to get this company like, okay, I'm going to vaccine company, I'm going to make this vaccine. So you get your product, whatever the vaccine, whether it's a live attenuated or the inactivated or with these new mRNA vaccines. You typically start off with a small group of people. This is called phase 1 trials. So this might be somewhere between 20 and 100 people. They're all volunteers. And so you're looking to answer these questions. Is it safe? Does it seem to work to do the job that we want it to? What are the side effects happening? And they're looking for dosages. How big of dose do we need to get the effects that we want without causing any dangerous side effects? So then after the phase 1, they move on to phase 2. So then the group is a little bit bigger, maybe several hundred volunteers. Again, they're looking to answer some of the same questions. What are the side effects, short-term side effects? Does it still work? How is the immune system responding to this vaccine? The dosage has probably been figured out by then. Then phase 3 comes around. So this is usually the biggest group, hundreds or thousands of volunteers, again, asking the same questions. Is it safe? What are the most common side effects? Is it working? Is it being efficacious to prevent the thing that we want to prevent, usually illness, symptomatic and severe illness? And then once phase 3 trials are done, then it applies for FDA approval. FDA looks at all of the research, all of the data that's coming in through all of these phases, and then they give the approval. And once it is approved, and even in the developing stages, they are always doing quality checks. They look at the lots, and they're always checking the manufacturing, making sure everything is being prepared properly. Currently the 3 vaccines that are approved in the United States, the Pfizer, the Moderna, and the Johnson & Johnson. I don't know the sizes of the phase 1 and phase 2, but phase 3 had anywhere between 30 and 40,000 people involved in that phase 3 trial, where in each phase 3, half of the group got it, half of the group was a placebo. And so then they can compare and give us that efficacy. How well does it work? And that's where we were hearing those like 95% effective against COVID for the mRNA vaccines. I think by the time Johnson & Johnson came out, it was maybe 70 or 80% effective, but variants had come into the system at that time. So this is the path that vaccines take. For all three of those, they were able to complete all of these phases, but before a full FDA approval, they applied for what's called emergency use authorization. So there was no skipping of these steps. A lot of people had concerns about that. And then the news came out just today that the FDA fully approved the Pfizer BioNTech vaccine. So that one has now full FDA approval. Okay, so now we're going to take a look at these COVID-19 vaccines. How are they similar, and how are they different from the traditional vaccines that we are more familiar with? Okay, so to take a look at what it is we're fighting, the picture on the left is a drawing of COVID-19. It's an mRNA virus. So what that means is its genome, its instructions to code for everything it needs to, is in RNA. It doesn't even have any DNA, so it's a little bit different than our cells. It is surrounded by an envelope. And on that envelope, what we are interested in is what's called the spike protein. That's that. In the other picture was that red thing. This is what allows the virus to recognize our cells and sneak into our cells by binding to our receptors. Okay, so its genetic information is in RNA. So again, I'm going to move you guys on my screen. I'm going to put you over on this side of the screen over here. So what I have in this box is the genome. It's like a map of all of the instructions on this particular virus. So it has about 30,000 letters, those bases, A's, T's, C's, and G's, about 30,000 long. We have hundreds of thousands of bases in our genome. So we can see some of those sequences, right? The little recipe cards are going to code for one particular part of the virus. The one that we are most interested in is the spike protein. That's this guy right here, kind of the blue and green one. It's made up of a couple different pieces. So in these mRNA vaccines, what researchers have been able to do is they can take, I'm going to call them scissors. They're molecular scissors. We've been using this for a very long time. They've been in genetic research for a long time. And what they've been able to do is they go in and they just cut out that one little piece of the entire instructions for a COVID-19 virus. They just want the instructions to make that spike protein. So that's the main ingredient for your mRNA vaccines. Okay, so for Pfizer and Moderna, I'm going to scoot you guys back over there. That piece of mRNA that we just cut out, right? That's this guy right here. Researchers identified that way back in like January, right after January of 2020, I should say, right after COVID kind of came on the scene. Researchers already identified that sequence for the spike protein. They started making they had the vaccine ready to go by March of 2020. They started phase 1 trials that soon, just within a few months of this recognition of that sequence for the spike protein. So what's new about Moderna and Pfizer is this mechanism of how that spike protein gets in the body. Traditionally, we would have cultured COVID-19 viruses for a very long time, trying to keep that spike protein but get rid of their pathogenicity, or to break it up and just put the little bits in a vaccine. And there are still some people working on vaccines in that traditional way. These new mRNA vaccines, they're going to use how our cells normally make proteins, and they're triggering our cells to make that spike protein. So they take that piece of mRNA, they put it in a little fat bubble. It's called a lipid nanoparticle. It's just a tiny, microscopic, tiny little piece of like a fat bubble. They put the pieces of mRNA in the fat bubble, and that's the vaccine. That's really all it is. Pieces of mRNA that code for that spike protein wrapped in a little fat bubble. That goes into your body, right? So this is your injection. The fat bubble fuses with your body cells because our body's membranes are made out of lipids, similar types of molecules. Now that mRNA is inside of our cells, it lands on a ribosome, right? We're making our cookies. It lands on the ribosome, it reads just that mRNA, it manufactures a spike protein, right? So here's the spike protein. So our cells are making the viral spike protein. Here our body recognizes it, activates our immune response, and now we have antibodies, okay? Both Pfizer and Moderna, are in the same method. This is how both of those mRNA vaccines work. Johnson & Johnson is very similar, but a slightly different delivery system. So in Johnson & Johnson, these are called viral vector. I think the AstraZeneca one that was approved over in Europe follows this same path that Johnson & Johnson did. So they do the same thing, is they're finding the spike, the gene for the spike protein right here. Oh, red on pink doesn't really work. There we go. And instead of wrapping it in a little lipid nanoparticle, they insert it into into another virus called an adenovirus. It's harmless. It does not cause any illness, but it's a good messenger. It's a good delivery system. So instead of a lipid nanoparticle fusing with our plasma membrane, this virus basically infects our cell and it inserts that piece of RNA into our cell. And then from there, it functions the same way. The piece of mRNA lands on a ribosome, making the spike protein, and then our body's immune response recognizes that. So in both of these mechanisms, all three of the currently approved, either the fully FDA approved Pfizer, the emergency use authorization for Moderna and Johnson & Johnson are all three mRNA vaccines. The major difference is the delivery system, either a lipid nanoparticle or this harmless adenovirus vector that delivers the information to our cells. And then our cells make the spike protein. That's the new part to these vaccines. But the science behind this has been in use for decades. And that's it. So that is my presentation on our cells, our immune system, and the COVID virus and vaccine. So I am happy to take questions. And I can go back to any of the slides if you wanted to review any of the pictures. Hi, Dee.
Hi. I know there's been no formal decision on this at the government level, but have you heard anything on booster shots recommending that you get a different type of vaccine than your original shots?
I've read a few things, so I try to keep up on the articles that are coming out just because I'm curious. It's kind of the nature of, my lifestyle. I love science. But there have been some studies of mixing and matching, like if you got Pfizer first to get Moderna, if you got Moderna to get Pfizer. I don't know if Johnson & Johnson's a mix and match in there. But at this point, they have recommended boosters, a third shot for Pfizer and Moderna, not for Johnson & Johnson yet. I read the data is still kind of coming out on that one. But at this point, they're asking you to get the same one that you got originally. It's open for immunocompromised people at this time. And then starting September 20th, I want to say 8 months after your last shot, you can get this booster. So for me, I got my last shot, I think in May. So I'm not due until like January or February or sometime. And who knows what the situation is going to be like then. But I don't know if there's been any peer-reviewed published work on the mix and match between the different vaccines.
Thank you.
Mm-hmm. Any other questions? There's so much new stuff coming out all the time as well. So, whatever we are talking about now, it is going to be changing as new data comes out. But since these vaccines came out in January, hundreds of millions of Americans have gotten them with really, really good outcomes. Even though there's a few breakthrough cases that are happening, most of them are asymptomatic or very mild compared to a full-on COVID exposure.
I have a question.
Sure.
So when we hear that there's all these variants out there,
yes,
and they could or could not make the vaccines work, what specifically is different about it? Is the spike protein altered or?
Yes, very good question. So I'm going to go back to this diagram of the gene. So mutations, these variants are caused by mutations, and it's a random event. So we have mutations that happen in our cells as well, but we have ways to kind of repair that. And so we don't get a lot of problems. But viruses replicate very quickly, and they don't have as good proofreading mechanisms as our cells do. So let's say there's a mistake right here in this gene, right? So I'm going to draw this line. So that change in a letter, an A, T, C, or G, whatever, that maybe it's going to make this part of the spike protein have a slightly different shape. Maybe it's going to have a couple bumps on it instead of 2 bumps. Now, these mutations are happening all of the time, and the more infections there are, the more chances of mutations kind of being picked up. And there's a couple different ways, couple different things that could happen to that mutation or to that particular virus that has a mutation. One, nothing could happen. It's not going to impact the functionality of the virus at all. Second is it could make it less infectious. It could be detrimental to the reproduction of the virus. And those mutations typically die out in the population, because if your virus can't infect, it's not going to pass on that trait to the next generation. The one that we are dealing with is if that change, that random change in the gene, makes that spike protein or any of these other proteins altered just enough that increases its ability to be infectious, that's what we're dealing with. So from all of the variants that have come out, the first one was alpha, beta, gamma. We're dealing with delta. So whatever that mutation was, again, randomly occurred in some virus somewhere. I think it originated in India this time. It changed the shape of this spike protein that made it bind to those cellular receptors a little bit better, and made it replicate a lot more efficiently. So it's way easier to transmit the Delta variant than the other previous ones. So some of the numbers I've been hearing in the original Alpha variant, one sick person infected about 2 to 3 people around them. The Delta variant, one sick person can infect anywhere from 5 to 9 people around them. So the contagiousness is what changed with the Delta variant. Whether the severity of the illness has increased, there's this, they still really don't know about that. I don't know if there's enough data to come in to say if you get Delta, are you going to get sicker with Delta than you would have with the original COVID or with the Alpha variant, the first one to come on scene. They don't know that quite yet, but we're going to see more mutations, more variants come out, and how they have an effect on the population is all about how that spike protein or any of those other parts of the virus have changed enough to either transmit easier like Delta, could be more damaging to our bodies, which would make more symptomatic and severe illnesses. The big question is, are we going to get a variant that's going to come along that is not recognized by your immune response? And that's kind of what happens with our yearly flu. So the flu mutates a lot. And so that's why every year we have to get this updated flu vaccine, because our cells aren't going to recognize, our memory cells are not going to recognize those new strains. And so the big question is, are we going to see a COVID variant that's going to come along that people who've recovered from COVID or people who've got vaccinated with these vaccines, will we be able to recognize, or will it be like a brand new infection again? That we don't know. It could, but that's one of those big question marks. We just don't know, but that's the mechanism of how those mutations work. There's a change in that instructions, and the protein spike, just it's physically, that 3-dimensional shape is different. And usually the mutations, the variants that are successful have given it some kind of benefit or increase in efficacy over any of the other strains. So that's why Delta now is like 99% of all infections, is because it just does a better job of infecting humans than any of the other strains have done so far. Did that help? And did that kind of make sense?
Yes. Yeah,
Okay.
that was perfect. Thank you.
Oh, you're welcome.
Well, that sounds like that might be it. Great presentation.
All right, thank you, Sean.
Absolutely. And we'll see, closer we get to fall term, I'd assume we'll probably repeat this once we get a few more students on campus too, maybe even into the first week or 2 of fall term.
Okay. Yeah, and I do. So with this recording, I'm going to process it and I'm going to send it. I already have the PowerPoint posted. And then I had the video from January posted. I'm going to have Felicia Jenkins update it with this one, since it has a Johnson & Johnson information on it. So
Nice.
it also will be available on the LibGuides. And so you can email her if you can't if you don't know where to find it, but that resource will be available. This will be available as well.
Sounds perfect. And thank you.
Okay, are we done?
I think so. Last call.
Okay. Thanks everybody for coming.
you.
I hope you learned something and share the knowledge.
Thank you, Corey.
Yep, bye.